Welcome everyone to our bioelectrodynamics webinar. Before we start, I would like to give a few housekeeping notes. So typically we have our lecture about 45 minutes and the attendees are by default muted and not shown. But if you have any question during the talk, please don't hesitate to raise your hand or write the question in the chat, and I'll try to communicate it to our speaker. Anyway, after the talk, we'll be pleased to have a question and answer session, so please don't hesitate to join that. So I understand, just to double check with Professor Praprotnik, Matej, so do you agree that this talk is recorded and if everything goes fine, hopefully, then we can also share it on. So is it okay? That's just double checking that.
It's okay. If we manage to get it recorded, by all means.
Yeah, fantastic. Thanks a lot.
Okay.
Good. So I should also mention that this webinar is supported technically by International Union of Radioscience, as well as by Engineering in Medicine and Biology chapter of the Czechoslovakia section of IEEE. And today we have a great pleasure to introduce our speaker, Professor Matej Praprotnik. And he's the head of the laboratory for molecular modeling at the National Institute of Chemistry in Slovenia, and also he's a professor of physics at the Faculty of Mathematics and Physics at the University of Ljubljana. He's a physicist by education. Both his bachelor and PhD degrees are from physics, as far as I understood from your biography, correct?
That's correct.
Fantastic. Then he carried out his postdoc research at the theory group at the Max Planck Institute for Polymer Research in Mainz in Germany. And Matej also served as the president of Slovenian Biophysical Society for 5 to 4 years until 2019. And very interestingly, and congratulations for that, still not too late, that you've been a recipient of the ERC Advanced Grant 2019. And also he's been a coordinator of European Research of the project coordinator at the multi-X scale, the Euro HPS Center of Excellence. And as you will see, Matej's research is focused on computer simulation of soft and biological matter, and the focus of his research is on developing and combining innovative computational and theoretical methods augmented by machine learning techniques to study complex molecular systems. So we particularly asked Matej to give us some of the insights of his recent work on subterahertz excitation of proteins, but I would like to give now word to you without further ado. Please go ahead. Oh, I think, but I see stuck, so it works so far. So let's see if it goes. We got a speaker screen frozen, so hopefully he'll recover and can start. Sorry for the inconvenience. He probably found out the screen got stuck, but hopefully this was only single error, and hopefully it will not repeat. Let's wait for a speaker to get back. Hello, Matej. Great to have you back. Sorry for the inconvenience. Hopefully it will work further on.
Okay, I will try one more time, right? Share.
Thanks for the patience.
No problem. Okay.
Okay, looks like it's lost the speaker again. So let me see how the solutions we can find. So,
okay, now it should work. Let me try.
Okay. Okay. Great. We do see your screen.
Okay, so maybe now after.
So let's give it a try.
Yeah. Yeah. So it should work.
Yes, please go ahead.
Okay, sorry for this inconvenience. Thank you very much for the kind introduction. I'm very happy actually to present our work at this webinar, because somehow I figure out we work in the same field, and actually I will tell you today a story how I enter into the field, because I'm coming, let's say, from a completely other field somehow. Okay, it's connected to multi-scale modeling and soft and biomatter, but let's say from atomistic up to continuum, and actually this whole project is then connected more or less to length scales and time scales above that. And we are currently working on new models and methods that are connected to my ERC Advanced grant, and this is just, we are about to submit some nice work, but this is still, let's say, an overview of how everything connected for me to get to to enter this field of ultrasound guided target drug delivery. Okay, so the title is sub-terahertz acoustic excitation of protein motion, and basically the whole presentation will be how to actually put together all the methods required to study such phenomena, and also then going to larger length scales. I will start with the definition of some basic definition that we will know what I'm talking about. From the thermodynamic point of view, so from the statistical mechanics point of view, we are distinguishing three types of systems. First, there are isolated systems, which they have no exchange of matter energy with surroundings. So these are typically modeled or studied in microcanonical ensembles, if we are talking about equilibrium situations. Then we have closed systems, which exchange energy, but they don't exchange any matter with the environment. And that means that actually they have a constant number of particles. And this means that the typical statistical ensemble for this kind of system is canonical ensemble. And then we have open systems, which they exchange matter and energy. So that means that the number of particles changes, fluctuates in typically, and basically in thermodynamical equilibrium, we modeled such systems in ground canonical ensemble, if we are talking about equilibrium, of course. And today's talk, open systems are basically the most common in nature, and today's talks will be mostly devoted then to open systems, which is actually important for all the ultrasound modeling later on. And today's talk, this is the outline of the talk. I will first explain our methodology, and then I will showcase this methodology on some biomolecular applications, from DNA molecule in salt solution, then liquid crystalline phases of DNA that you find in our cells, in our or in viruses, for example. And then I will tell you how we applied this methodology that is basically developed in the context of multi-scale simulation to these biomolecular systems. We will go to the non-equilibrium situation, like rotation dynamics of protein under the shear flow, and then we end up with the ultrasound, basically propagation through liquid water, everything in the context of molecular dynamics simulation. So these are basically particle-based methods that I will be talking about today. So this is maybe important for this context. Okay, so it's a talk about multi-scale methodology. So what do I mean by multi-scale modeling and simulation? In our case, this means that we are concurrently coupling different methods for different length scales at the same time within a simulation box. So there is this phase diagram, and basically on the left and bottom corner you have a molecular dynamic simulation of a protein embedded in water, where the nearby, so the protein is now represented in a cartoon fashion, but it's basically an all-atom model of protein. It's, I think, some very easy, and so basic protein, I don't remember which one now, ubiquitin, I think, and it's embedded in water, and water is where it's important. There is this interaction of protein and water in the first closed shell, so hydration shells around the protein. The water is there represented using atomistic, full atomistic detail, whereas far away we can represent water on a much coarser models. In this case, here is just a Martini force field. So every blue bead represents 4 water molecules, 4 atomistic water molecules. And the idea here is that basically this is an all-atom, so it's a concurrent multi-scale coupling. Water molecules are moving around in the box freely, and depending on their distance from the center of mass of the protein, they change their resolution. So they go from 4 atomistic water molecules in the vicinity of protein into the one bit, and vice versa. And this is quite challenging from the technical point of view of MD simulation, how to exchange this resolution on the fly. So, and it's very simple to imagine how to go from 4 molecules to 1, but it's a little bit, the challenge is actually when you go in the other direction, when you go from the coarse grain description of water to more fine grain description, because you have to basically introduce missing information. You have to somehow reintroduce the atomistic degrees of freedom, and the more molecules 1 bit represents, the harder it is. But okay, so we managed to, I will present today how we do that. So this is one ingredient, and basically this is a multi-scale description. The point is that you need this high resolution interaction between protein and water only in the vicinity of the protein. So you don't need the whole information also for the distant solvent. And the point is that from the computational point of view, the most of the time, computational time goes into the direction, into the simulation of water. Even though you are basically interested into the dynamical and structural properties of the protein, and its which actually then determine its function, you are wasting your time on simulation of water, and this is one way how to speed up a simulation. And now okay, you can go then, you can abandon basically your particle-based description of solvent, and you go even to computational fluid dynamics. This is this middle figure there, where on the right you model that part using Navier-Stokes equation instead of Newton one. Basically, Navier-Stokes is just a mesoscopic version of Newton equation, and so it's quite natural how to couple these two descriptions. There are different strategies to do so. One is state variable coupling, where you have a overlapping region in between, and then you map somehow state variables like density, velocity of the solvent, so that in both descriptions they are the same. While here we actually use the flux exchange, where we are basically matching mass and and linear momentum fluxes. Between both description also you can match energy if you need to have this information in. And in this way, basically with this flux exchange, you can couple the both description of the solvent. And of course, computational fluid, for the computational fluid dynamics one, you can use some finite volume or finite element methods, engineering methods, or even Lattice-Boltzmann. It doesn't really matter which method you use. Here we use finite volume, for example. And then if you go even further up in this coarse graining, you can go into the nanofluidics regime. So here what you don't see, maybe what is presented is actually water flow through the nanotube membrane. So it's a membrane of nanotubes, and it's the idea was that basically you want to have so there was this pressing issue of drinkable water, and you want to desalinate water, so to get all the ions out, and you get just pure water on one side of the membrane. And these carbon nanotubes are extremely hydrophobic, and and from the energy point of view, this would be the idea is that this would be like a way how to desalinate water. It was a pressing hot topic, let's say 10 years ago in HPC environment. So here you just see 1/4 of one nanotube, but basically because of the periodic boundary condition and using the symmetry, this should represent the whole membrane. And what we did here is actually we did everything with the computational fluid dynamics method, even though one would have to use ND to compute this flow through the membrane. But what we did is actually we just needed one extra parameter, which is slip length between the water and the carbon nanotube. Basically, we needed to define the boundary condition of this interaction of water with the solid state structure. And one parameter was enough to reproduce the whole MD simulation, which were really HPC simulation at that time. And we could do this whole simulation on a laptop, basically, and we could reproduce all results. So that was the idea. But the idea is here that when you want to cover large time and length scales, you have to resort to, let's say, cheaper, less accurate methods. So ideally, you would have everything done quantum dynamically, but you cannot do this everything quantum dynamically for larger systems because your computational power is not high enough. And then you do this algorithm in the algorithmic way. So basically, with these multi-scale methods, what you do is you use the high resolution methods only in the domains of the system where this is really strictly required. And everywhere else you use cheaper methods. And by this clever concurrent coupling, you can then algorithmically speed up the whole simulation dramatically if you do it in the right way. Of course, this is not a general prescription. You cannot apply this methodology blindly to any systems. It just can be applied to certain subsystem in the whole realm of all systems. For example, you cannot compute all the properties if your coarse grain model does not allow you to do so. So, for example, if you want to compute the electric constant of water, you cannot use the coarse grain water because it does not have a dipole and a rotational degrees of freedom, and this is just not feasible. No? So basically, I'm just warning you that for certain subclass of problems and certain subclass of properties, you can use this kind of methodology, and in this way you can dramatically improve your simulation protocol and reach for length and time scales that would not be feasible if you just use the high resolution method. Okay, so how we couple? So in our case, we started with this coupling of a fine grain and coarse grain solvent within the context of MD simulation. Now, molecular dynamics simulation is just a method where you solve Newton equation of motion for all the particles in your systems. And there are many different computational codes, and this is a long and powerful method. that allows you to study biomolecular systems. Basically, you get all the information from the atomistic scale. Now, of course, it's a very, because these systems have a large number of degrees of freedom. It's also very time-consuming, and well, that's why we are developing this kind of multi-scale strategies. And in our case, we couple a coarse grain model on one side of the simulation box of the same fluid. In this case, some, let's say, artificial toy model, metal-like fluid with 4 atoms arranged in this tetrahedral structure arrangement, represented in this red on the right of your screen, and then the same coarse grain model of the same liquid, but where one molecule is represented just with 1 bit. And then you have to use some coarse grain interaction between molecules on the left side in order to mimic the, let's say, fine grained model on the right side. So this can be done then in different ways. There are these many different strategies how to coarse grain. Basically, you have to get to the potential of mean force ideally, but potential of mean force is many body potential. So if you want to do this in the pairwise fashion, like done here, you need to do some approximation, and you can then decide whether you want to reproduce the structure or you want to reproduce some other properties like free energy kind of properties, salvation properties, and so on. So if you want to do the structure, there are many different methods like iterative Boltzmann. Nowadays, many of these tricks you use basically machine learned potential, but machine learned potential are many body. Okay, so that's the only difference. So here we are still focusing just on the pairwise potential in order to speed up, because we are using this for biomolecular simulation. Here really we are focusing on sampling properties, not really accuracy so much. Basically we couple this with this force ansatz down there. This is a linear combination of atomistic and coarse grain forces, coupled with this weighting function that defines basically in which part of the your system a given molecule actually is at a certain point of time. So the weighting function 0 means that this is that the molecule is actually, it's like an order parameter. So it means that you have to switch to the coarse grain description, and if the weighting function is 1, you're basically in the all atom part of your box, and in between we have this hybrid region that basically serves the point. The point of the hybrid region is that, as I say, the coarse grain model does not have any rotational degrees of freedom, so, and also no orientation, so it's isotropic. And basically, when you go to left to right from the coarse grain to, so when you go from atomistic to coarse grain, it's completely straightforward. There you don't need any hybrid region. You would just have to throw away information, average out your rotation and so on. But the problem is when you go to left to right. So here you have to suddenly introduce rotational and vibration degrees of freedom that are not present in the coarse grain model, because it has only translational degrees of freedom. And in order to equilibrate them and find the right orientation and prevent the overlap with other molecules, surrounding molecules, you basically need this hybrid region, and then you slowly switch on the atomistic degrees of freedom by increasing this weighting function, so that when the molecules then enter into this atomistic region, they are already equilibrated with the right orientation and equipped to an external environment by themselves. So this all happens on the fly. And actually, just to mention, you need also a thermostat that provides this or removes this excess rotation, so the energy, free energy associated, and entropy basically associated with this rotational degrees of freedom and vibrational degrees of freedom. And the point of the whole, and then we have also this thermodynamic force on the right. So the point is that you want to simulate a homogeneous liquid. So the density profile across the whole region simulation box has to be constant. But because you're using different, you use fine grain model to simulate the same liquid in one part of the box and coarse grain on the other one, these two models, they don't need to have, so there is no reason why they would have the same chemical potential. And that means that the molecule will go into the domain with lower chemical potential density, which is typically the coarse grain one. So you will get you will end up in some stationary state, but this will not be a homogeneous density. This will not be a state with a homogeneous density profile across the whole simulation box. And in order to actually oppose this artificial chemical density gradients across the box because of using different models, we have this thermodynamic force there, which is basically the negative gradient of the chemical potential. And basically, in this way, this allows you to couple quite different liquids in the same model. So here we are using a coarse grain and fine grain, but you could also couple, for example, different water models, TIP3P, SPC, or whatever kind of models, because with this thermodynamic force, you can just oppose their chemical potential difference, and you will end up with the uniform density profile where both models are in the thermodynamical equilibrium. Okay, so this is basically this method. And then you can simulate different stuff. So here we can use, okay, so you see, you can use also different geometry. So here we have two boxes of the same, they are the same left and right. It's just a flat basically boundary between them, but you can also use cylindrical geometry, and you would use, in the middle we have a double-stranded DNA, this blue, it's all atom DNA, one pitch. Basically we use periodic boundary conditioning from below and up, so that the upper part sees the lower part, and basically in this way we have an infinite strand of the double-stranded DNA embedded into the sodium chloride solution. And then farther away we use, like I said, for water we use these gray bits, 1 bit per water molecule. And then you can, with this kind of multi-scale strategy, you can compute, for example, dielectric properties of the DNA for different parts of DNA molecule. And here above, what you can do is basically, as I said, the coarse grain water does not have any electrostatics, explicit electrostatic, because there are no charges, no dipoles and anything. And then you can basically, what you can check is how large actually this atomistic region that surrounds the DNA molecule has to be in order to, because of course the DNA, so the water molecule in the coarse grain part does also not form any hydrogen bonds or anything. So you can check basically by increasing this domain of the all atom solvent, you can basically check how large this, how far the influences of hydrogen bonding and electric charge is, so that everything stays the same as in the all atom simulation. And basically these numbers in the brackets, so basically this notifies the radius of the all atom region. So the larger it is, and infinity means all atom. All atom, the last column is basically reference values that you should achieve if everything is right. And then, of course, you can also calculate, as I said, the electric constant of water, and this is what you see here. Basically, the red curve is all atom results. And you see that the DNA molecule, because of its presence, it influences the value of the dielectric constant in its vicinity, but farther away, the water reaches the bulk value of around 80. This actually depends on the all atom model that you use, but let's say that it's all 80. And the other colors are basically this hybrid all atom results. And you see that basically here at the right, everything goes somehow down, but this is actually what I warned you. You cannot use this model for computing dielectric constant of water, because the coarse grain water molecules, they don't have any dipoles. So, of course, and also no rotational degrees of freedom, which determine the value of the dielectric constant. But what is important is that here in the vicinity of the DNA molecule, your hybrid model gives the same results as the all atom one. That means that all the results in the all atom part of your multi-scale simulations are the same as in the reference all atom counterpart. And this actually proves that the multi-scale model works. And actually, then we wanted to use this methodology instead of just having one molecule surrounded with this multi-scale solvent. We wanted to study basically liquid crystalline phases of DNA. So we have in cells DNA is really densely packed because our DNA is 2 meters long, and then it's packed in our cells, in the nuclei. And basically, the question is, DNA is a highly negatively charged molecule. So how the heck can this condensate if everything should go repel each other? So they should really go away. So it's basically because of the salt counter ions. So we wanted to study. So what happens is that depending on this concentration of DNA, you can really go from this DNA condensate in our cells will go from isotropic, cholesteric, hexagonal to orthorhombic. They form all different liquid crystalline phases in our cell. So we wanted to study this phase transition between these phases, and to this end, we actually simulate this DNA. So you see that there are these circles. This is basically DNA. These blue circles are DNA pointing toward you. So this is a top-down view of such a system, and it's in this hexagonal arrangement, and around we use this coarse-grained solvent. There is no place to put this hybrid solution within between the DNA molecules because there is simply no place because they are so densely concentrated. And what we wanted to compute is actually osmotic pressure. So osmotic pressure is something it's a little bit more challenging to compute in MD than just pressure, but here we have this equation of states, and we computed it for different phases, so from hexagonal to orthorhombic, and for different concentrations, for different salt solutions, so sodium chloride and then sodium chloride with spermidine 3+ ions, because the point is that this condensation happens only in the presence of multivalent ions. This is actually what is relevant. So sodium chloride solution is not enough. You would not get a condensation of our DNA in our cells. Okay, this can be done with this multi-scale strategy, but you can go and start coarse graining this water even more. So instead of 1 bit to one, so that 1 bit of these coarse grained molecules, it represents one water molecule. You can have it, like as I just said before, you can use Martini description for this bit, and that means that it goes from 4 to 1 mapping. Okay, so you have 1 bit represents 4 molecules. And basically, for that end, you need to actually add to this address that I just presented, this adaptive resolution, also this algorithm swinger, basically that redistributes the molecules across the bit. So basically, you know, in water molecule, so liquid water forms this hydrogen network. So it has this tetrahedral structure. But the point is that these hydrogen bonds, they live on their lifetime is on a picosecond time scale. So they form and disassemble and so on. And at a certain point, some 4 molecules may be close together, or 5 water molecules, they are close together, but at some time afterwards, they might be just on the opposite side of the simulation box. And because if you want to represent them with 1 bit, you have to make sure that this 1 bit, that you know the information, which 4 molecules this 1 bit represents, and they are not the same 4 molecules across the length of simulation. So we need to have this algorithm swinger that basically distributes water molecules, all atom water molecules among the bits, that you have this information, and this is important for this coupling. But you can go even beyond Martini, and you can use this kind of supramolecular coupling for dissipative particle dynamics. Dissipative particle dynamics is another model, and okay, this is just a nice representation of this swinger algorithm. So water molecules are swinging basically, and okay, this is basically it's a very simple distributing like k-means or something like that. It's not, I mean, it's quite simple how to achieve that. It's not, you can do this at every update of your layer list or something. You don't have to do this on each time step, and it does not represent any computational difficulty. So, okay, what is important for our discussion today is basically this MD-DPD coupling. And again, like with Weierstrass, DPD is basically a particle-based model that reproduces the continuum fluid hydrodynamics in the continuum limit, and it's a special subclass of MD. So MD is basically what you do is, as I said, you have a force field, then you calculate forces as a negative gradient of this force field, and basically this is what you do. Now in DPD, you use three types of forces: conservative, random, and dissipative. And basically conservative are just this conservative part like in MD, but in this case it has this special linear repulsive form. And the other two terms, they just basically represent a thermostat. They satisfy this fluctuation dissipation theorem, and this is a dissipative particle dynamics thermostat. This is just to say that this is the DPD is a special MD method with a special conservative potential, so with a special force field and a special type of a thermostat. But otherwise, they fall into the same kind of methods. And the coupling, what we needed to actually, because DPD is important to simulate all these fluctuating continuum fluid dynamics phenomena, you need to actually make sure that the transport of linear momentum through the liquid is preserved using this coupling strategy. And that's why we checked that the linear momentum, this is actually local linear momentum across the x coordinate of the coupling coordinate of the two methods. Basically, the linear momentum transport to the liquid is goes well, even though we are coupling two different kinds of methods. And then you can use this coupling actually, as I said, also to couple it to the non-particle based models like computational fluid dynamics models. And this is actually useful, for example, target drug delivery. So you have your fluid flow past some, in this case was a buckyball, which is poisonous, but okay, it just served our example. So you don't know the boundary condition between this carrier and your fluid flow, because these guys are on these are nanoparticles, and they are not colloids. If they were colloids, you know the boundary condition, it's non-slip boundary conditions, but these are too small objects, and you have a partial slip, so it's no slip, it's not full slip, it's something in between. And as I said, the parameter that determines this boundary condition is actually this slip length, and you can compute it actually out of using a more accurate method, which is MD. So, but MD, we are not using in the whole box, we are just using it around the shells of this buckyball. And then you get your slip length, and then you have basically a boundary condition for your continuum fluid dynamics solver. So this is this hybrid methodology, and basically you see how you can study it two ways, how your flow outside influences your molecular region, or how your molecular region influences the flow. So it goes both ways, depends what you want to study. And actually, and then you see that if you just focus here on this particle-based part of this whole box, this is an open system. This is what I said at the beginning. Here, in this part of the particle-based box, because we have a fluid on here also in the continuum domain, the number of, the mass flows and linear momentum flows, they go from left to right, and at no time, so the number of particles in the particle-based part of the box changes over time, because it just follows what the continuum part of the box dictates. If it says the mass flow is running into your particle-based part of the domain, you have to increase molecules, otherwise you decrease them, and that means that this is an open system. And actually, then you just follow this particle-based box. You see that you can, okay, here we are representing, it's not water anymore, it's basically star polymer melt. Actually, it's a very dense fluid. And actually here, what we derive here is an open boundary molecular dynamics. So you need to have instead of, usually when you do the particle-based, so when you study liquids with particle-based methods, you use periodic boundary conditions. So when the molecule enters from one part of the box out, it enters in the other part. So in this way, you are mimicking infinitely large molecular system, but you're keeping your number of particles in the simulation domain constant. Okay? So now if you want to do the ground canonical MD, it's not very typical. So this is usually do the MD in a canonical or microcanonical ensemble, but not in the ground canonical one. And the point is that what's the problem? The problem is, you can see on this picture, actually all different colors represent one star polymer, and they are intermingled and it's very dense. So if you want to change the number of particles in your simulation box, you can do this in two ways. Either you reduce them. Now, deleting a molecule is not a problem. This you can always do. But to add one molecule into such a dense system, it's impossible. You have to now find a temporal gap that forms in your liquid and insert it there. And if you have such a large complex molecules, this will never fly. So in our case, we actually do this in this coarse-grained parts, which we call buffer on left and right. So now in this way, we open up the system in the horizontal way. And actually here we delete and reintroduce molecules, because these coarse-grained molecules, they interact with each other with much softer interaction as the fine-grained one. And basically, you can allow for certain overlaps within them, and then you use a dress basically to reintroduce the atomistic details, and then you can have an open system in this region of interest. So here, this is, and actually here we don't use periodic boundary condition anymore. Instead, we can prescribe any boundary outside. So it's like in an experiment, we can, you imagine that you have a box of water and do something weird on the outside of the boundaries with it. So depending what you do, you can introduce two, let's say, opposite pressure. You can actually represent, basically you can, we do this through this pressure tensor, and we can do many different things. And then what you can do is actually go back to this DNA system that we used before and say that instead of coarse-grained molecules on the outside, we just use an implicit solvent with the dielectric constant of 80. This is implicit. You just preserve ions. But then this all-atom solvent around the DNA molecule, because the DNA molecule, they go into continuum and back, this number of molecules around the DNA molecules is not constant anymore. And actually, this open boundary MD allows you to simulate this kind of a system. Now, what is good to this coupling to the implicit solvent? The problem is that all these biomolecular simulations are typically simulated using 1 molar source solutions instead of physiological ones, which are 0.15. So they are much lower. And what does it mean? Why this is the problem? So the problem is that if you have a low concentration, which is physiological, you need to have, if you want to have a certain number of ions to have an acceptable statistic, you have to have a large number of water molecules. And that means that your simulation slows down dramatically. So it's better to have a higher concentration of ions, and then you can afford to have smaller systems, so systems with smaller number of water molecules, and this actually, it was from the computational point of view, it's much easier to then simulate systems with higher concentration. But in our case, basically here, because we don't have any water here, we can go to this physiological concentration with no extra cost. So this is actually the way how to do all this simulation at the physiological conditions. And then if you want to get rid of the all atom water completely and preserve all the correlation between electrostatic correlation between ions, this is what we also did. We actually go to the machine learning. We did machine learning potentials where we trained our network. We use this Allegro code for equivariant neural potential. These are graph neural networks. And we train our coarse-grained model of the DNA plus ions on all atom DNA plus ion system. And then we remove the whole water. Okay? We remove, there are no water, but just ions and DNA. And actually, we preserve basically our structure and dynamics of the all atom model, but there is no water in our machine, in this machine learned potential, where the machine learned potential is of course between DNA and ions themselves. So this is our coarse-grained model that represents all the correlation. Now, with this open boundary MD, you can do different tricks. You can then do not only, you can go beyond the equilibrium situation, so we can put your protein, for example, under the shear flow, and you study this influences of the shear flow on the proteins. These dynamical aspects are very important for pharmaceutical industry, because by transport of these drugs, you can go through these channels and you have these, let's say, curves, and then they enter into this rotational dynamics. And since rotational dynamics, it rotates the protein, you get very and elongates the protein. You can get you can enter into some condensation phenomena that you want to actually avoid in pharmaceutical industry. So this you can simulate. You can simulate with this methodology. You can do all this, simulate all these kind of phenomena. But what is really important, you can actually use it for a virtual ultrasound machine using particle-based methods. So this is actually then a DPD model. So the DPD is now, it's very flexible, because here you decide how many molecules one bit actually represents, and this actually then determines your length scale where you are simulating all this stuff. You introduce your ultrasound as a boundary condition, as a basically harmonic perturbation, and then you study the propagation of your ultrasound wave, and you get we compare. So the blue points are basically our molecular, so dissipative particle dynamics data, and the black curves are basically reference continuum, continuum fluid dynamics data. Basically, you get attenuation of the sinus wave, as you should. And then you can use this kind of virtual ultrasound machine, for example, to excite protein motion. So we did here. We send this one plane wave. We basically shoot one protein, and we study the vibrational, the distribution of states. Actually, this V is this. It's a time derivative of the radius of gyration of such a protein, and we could see we use this. PCA, normal mode analysis, and basically checked whether we can excite normal modes with ultrasound. So in principle, we want to see if we can influence, principal component analysis of these modes by if something changes if we put the protein under the ultrasound motion. And indeed, in our case, we could show that basically we can do that, and you can see that this basically the dose of all these states is increased when we hit the frequency of the normal mode with our ultrasound. So that's like a oscillator, that's a forced oscillator in physics, in the simplified figure. So we use here a simplified model, Martini model, and we had just 6 bits. So we use 6 coarse-grained bits to use this to compute this principal component analysis. It means we had 18 modes in our case, and we studied that if we could basically what changes when you have ultrasound present, and if you don't have an ultrasound present, then we could really see that we can induce normal mode motion if we shoot with the proper, we choose proper ultrasound frequency and intensity. And this is actually important for ultrasound target drug design that we are doing now, actually. And okay, so I will also maybe conclude here. What we did also was we now introduce a framework based on this SIND, sparse identification of nonlinear dynamics method, where for these DPD liquids, we actually get out the Navier-Stokes equation. Out of the microscopic dynamics, underlying dynamics, we can derive the macroscopic model, macroscopic dynamic equations using this sparse identification of nonlinear dynamics. This means that we have to actually, you have to have a liquid, for example, in our case, you do the DPD simulation, and then you compute trajectories of some macroscopic fields, in our case, mass, density and two velocity fields, and then you basically choose a library of possible derivatives, spatial and temporal derivatives, and then basically we derive a model selection that leads then actually to you get Navier-Stokes equation out of that. So basically, this is the conclusion. You know here what you have. So what the idea is that if you have a dynamics that you don't know what the dynamics is, that you use this methodology and get the unknown dynamics automatically out of your trajectory of your system. Okay, so this is all now the basis of for my ERC project. Yeah, it's running some time, but it was some there. We had to overcome some difficult phenomena, so now we have a few articles ready, which will appear very soon, and this will actually scale up to more to larger systems like microbubbles and gas vesicles. And we have, as Mika mentioned, also a center of excellence, basically devoted to code development. This is code development of these existing codes. This all will appear, these codes that I introduced today will appear basically in open, public, accessible stash of these methods, which we develop here in the multi-scale. Everything will be available in lumps, and so for everybody to use. Here, actually, we also, this multi-scale is not only devoted to the codes connected to this ultrasound biomedical application that I presented now, but also to batteries and even helicopters. And they are all basically they are coupling this particle-based hydrodynamics with continuum hydrodynamics. This is the, let's say, a common denominator for quite different physical processes, let's say, and phenomena, but the code-wise, they are similar. Okay, and here I would like to acknowledge my collaborators. These are my collaborators and students, and also ERC and Slovenian Research Agency for funding, and you for the attention. Yeah, okay, thank you.
Thank you very much. Very interesting, both methodologically and scientifically. And it's really, as you mentioned, in a way synergistic yet complementary to our approaches, because we've been most looking on the things from the perspective of, let's say, electric field response or oscillating electric field response to of our protein systems. But in earlier past, I think when I was still a PhD student and a young PI, we also had a look on let's say, vibrational phenomena on a molecular scale, and we often have questions which relates to your actually methodology. Mm-hmm. So I have a couple of questions. If,
sure.
Let's see if anybody from the attendees also will raise the question, but let me open my notes. So I wanted to ask, thanks for defining the what you mean if thermodynamically isolated, closed, because sometimes these are used, loosely. But now thanks for the definition as you use it. I'm wondering if you would, for the particular work you did on a subterranean excitation of protein, do you really need there having the system to be open in terms of exchanging the water molecules from, let's say, one domain to another one?
Now here, basically, okay, so In this case, I think it's more to this connected to replacing periodic boundary conditions with something else. So
I see. This is how I got it. Yeah, I see. I see.
So in this way, basically, the idea here is, you see, you have methods that are known for this non-equilibrium MD. You have shear MD and so on. So you have slot. There are many methods, and the way how they are devised is you change your equation of motion. So you add to your equation of motion, which are Newton equation of motion, whatever they are, certain extra terms that drive your system to a new stationary point, which is like this shear stationary point. The bottom line is that you need to know what your stationary state is, and then this extra term drive the system there. So in our case, it's a little bit different. We are getting to this non-equilibrium situation by prescribing our external forces on our system. Okay, and this can be, you are free to do so. You don't need to, you can do something completely crazy and your system will not reach the stationary point. You don't know what the stationary point is. But basically inside of this region of interest, inside of the molecular region, we are not adding any extra term to the dynamics. So it's completely Newton dynamics. The whole thing just adapts to what you are doing it on the outside. So you are much freer to do, I mean, it's much more simple to do many different things that with the standard methods, because you are much more free to do so, because you don't need to change your equation of motion. The molecular system within will adapt to what you are doing it on the outside.
Okay, I see. Okay, thanks. There is one question, Saurabh, if you mind asking it. I unmuted you, and you can also unmute yourself, Saurabh, and ask if I don't know if your microphone is working. If you give it a try. It's always more fun to have people directly speaking than just reading. Anyway, so if you, Matej, can read the chat, or I can read it for you. There was a question from one of our team members. He's also MD simulation guy. Can you see the chat here in the?
I cannot see the chat, and I don't want to touch anything to not.
Okay. Anyway, so I'll read it. So Saurabh was asking. Well, saying first, very interesting talk. Thank you. Do you see any difference in the dynamics of counterions in a coarse grain versus all atom part of the box? For example, in the simulation of DNA, do the ions tend to stay longer in atomistic water part? I mean, this is even implicit water, right? We didn't go farther away.
Exactly. So here we use, so basically the dynamics typically in this coarse grain model is different than the all atom one. That's because you are basically avoiding this shear. No, I mean the interactions are much smoother, and you are basically getting away from all the internal viscosities. Okay, and you have to somehow reintroduce that. Typically, this is not done in this coarse grain models. You are concentrated only on structure. So then you are they are much faster, typically, dynamics in the coarse grain model. But here, what we showed, actually, we were focusing on this, where was it? No, maybe not there. So what we were focusing was basically this times of residence times across around the DNA, and they were comparable with both.
Okay.
Because actually, this is what I wanted to show. We want to have everything the same in the all atom part of our box. Everything is the same as in the all atom simulation all over. Okay, so we are not really focusing what's going on here in the coarse grain part. We are not interested. We are interested in this case that we can get everything right here at the vicinity of a DNA molecule, and this is true. So the dynamics of the all atom water in our hybrid model is the same as as the all atom in the full all atom simulation.
Okay.
But I agree that the dynamics in the coarse grain part, it's not the same. So, but we are not interested for.
I see.
Yeah.
I see. Okay, so I was wondering about something which is my related two interrelated questions I have regarding the question of effective slip at the interface of the protein and water in terms of driven when you drive that protein with the external, for example, external pressure oscillations, and it kind of relates to effective. What would be? Have you tried to extract something like effective damping coefficient for this purpose for driven protein vibrations?
This figure,
for example, this one, or yeah, it's a little bit complex, but yeah. First, let's make it simple. If we have a simple shear which is not oscillatory, just
this one,
continuous one, yeah. So can you speak something about as a slip, or is it a inappropriate concept for and on this scale?
Slip and no slip, as I said, these are too small. This is if you want to have this is actually partial slip in principle.
Okay.
Because in these small regions, also here, when we have this kind, these are even, this is what, 1 nanometer, radius is 1 nanometer around, I think. So this is comparable, and that's the problem. So they are too small to be like colloids. Colloids are 1 micrometer, there you have no slip. And this is actually somewhere in between. You get this slip length. So here we computed here actually the slip length out of the velocity profiles.
Okay.
We fitted the slip length, and then
so you got like effective slip length for this case, although I know it's okay.
And that's actually, this is what I show in the first figures. So here in this the upper nanofluidics. Let's say flow through the nanopores. Okay.
Okay. Here that was the connecting parameter. Yeah, I see.
This was this missing parameter, and this missing parameter you could then reproduce this flow enhancement. It's called. So you compare it to the Poisson flow of the normal pipe, and that's why this carbon nanotubes were so interesting, because you got this normal fluid flow enhancement, and basically we could reproduce with just one fitting parameter. This is this slip length, which is again, it's a partial slip. You could get all the MD results.
Okay, that's very interesting. Okay.
Of course, you need to have large enough radius. I mean, radius should not be too large in order not to go to full slip, of course. And but not too small that you would need to include, for example, quantum mechanics, because you don't have this one line of these water molecules. Now this is larger, but I don't remember how large the radius was, but basically there was just a few streamlines, not more. So this was this fluid, let's say Navier-Stokes, works pretty well also on this molecular scale that basically, in principle, you would not expect it to work. But the continuum equations are quite powerful, so you just need a few streamlines and you are already in continuum regime. So this is but of course you need to have a boundary condition.
That's interesting, because we've been trying to understand the, let's say, nanoscale vibrational mechanics of polymers such as microtubules, and since the 90s, now my past supervisor, he was kind of predicting that what if there is because people typically consider to be a stick boundary condition, so that caused a lot of damping in terms, even you wanted to drive the structure because you were using the energy to the stick boundary conditions. But they were saying, what if there is a partial slip? So we only introduced it in, well, already 10 years ago as just an arbitrary parameter, but we didn't have much of the arguments for it.
Of course, you get this, let's say, friction force is between 4 pi and 6 pi prefactor, right? It's 6 pi is no slip, 6 pi r v. This is Stokes formula. This is no slip, and then 4 pi the pre-factor 4, it's 4 or 6, and this is there is a formula, so we actually put it. I know that we have it somewhere in our article. So the point is, with the slip length, instead of infinity or 0, you get from 4 to 6, and if you don't have it with 0 or infinity, you will get the value disparsely between 4 and 6.
Okay, I see. Maybe we can get back to that maybe in another discussion, but just to move forward. And okay, see if anybody else. So okay, so what are the shear flow effects on protein? For example, if you drive protein in some flow, can it cause protein for reasonable flows? Can it perturb the protein structure?
The shear has 2 components, right? So shear flow is half elongation flow and half rotational flow. You get all this coupling of rotational and elongation dynamics. You get this, it's similar to the red blood cells. You get this tongue threading and tumbling dynamics. So basically, you can study. So it's a rich zoo of phenomena here. What can be done, and basically the way this influences actually on condensation of proteins.
You mean aggregation?
Aggregation, sorry, not condensation. Aggregation, and this is actually where the pharmaceutical industry is actually interested in. So because they want to prevent the aggregation, because then you lose the drugs efficacy. Yeah.
I see. It's interesting. Interesting. I was trying to understand how does it relate to the case, for example, when in our case we drive the protein motion through the liquid by electric fields because the proteins are charged, at least those which we use. And it's not a shear flow, but it's definitely a flow.
That elongation flow that it. So here they start breathing. You have this cyclic motion. They can become large and then they shrink and so on. So it's because you have this shear has these both components of the flow, so rotational and elongational one.
Okay.
And so then it depends. It's a large phase space, so you can. It depends on the shear rate and so on. And the frequency where it's connected, and it's very rich phenomena. It's not only for proteins, you have it in all kinds of polymers as well. So it's very interesting from the point of view, from the physics point of view, because it allows you to it's a complex thing, and you can really study and try to understand and connect different dynamics to different kind of motion within the polymers. So you have different characteristic times of polymers, and then this is connected actually to all this kind of phenomena. So it's I cannot just say it for everyone because it depends on your polymer and many things. But in principle, it's a rich. It allows you a rich phase space to study.
I see. So probably one of the things people will be interested in a lot is the frequency selectivity of these excitation by sub-terahertz oscillations. So could you see in these responses that the vibrational response is something like underdamped? Because typically people say that at these frequencies at tens of gigahertz, 0.0 something of a terahertz, that the vibrational motion is overdamped. But is it the...
So we studied this now in the latest paper. Now I don't remember. So we wanted to have what is connected to the temperature or not, and so on. If you have so the attenuation definitely depends on the frequency very much.
Yeah.
And here, for our case, this is important because here we are now, it also determines the length scales that you want to study. So here we are talking about the individual protein molecules, and we are in this sub-terahertz regime. So in our case, in the other part of our ERC, we are going to megahertz, basically.
I see. Yeah, that's well you can...
It's much larger. And the point is that here, basically, this what I showed, this figure, you see that this wave will attenuate on a nanometer scale. So if you want to study something on, let's say, centimeter scale, like they do in biomedical patient, you will ask yourself how the heck they can reach centimeters if everything dies already on the nanometer scale, because this attenuation
that was my question.
is basically frequency dependent. So yeah, okay. So if you want to go to the higher length scales, you have to also change the frequency. And this is the point. Ultrasound is, you know, it's a very vague definition. So basically everything above the 20,000 hertz. I mean, this is a vast area of frequency range. So, and then that is actually connected then to the length scales, yes.
So one thing is just if you consider the acoustic waves, one thing is the attenuation. That's of course clear. Then even generating the sub-terahertz oscillations or acoustic waves will be not easy feat to do in the water. I can imagine it's possible. But for practical applications,
we are talking here about modeling part. This is already difficult. So the experimental part is also not a piece of cake. No,
yeah. But what I didn't mean now really attenuation in terms of propagation through the material, but in terms of the damping of the protein vibration as an oscillator. Like
this is something. Okay, so here I have to say that this was the toy model of the protein. This was. Ubiquitin, it's a simple molecule, but it was also a Martini force field plus elastic network. So we didn't have a completely, but this was not because of it was actually because due to the code. It was our code that we used at that time was, we could not allow to have, let's say, more refined models and larger molecules. So now we are repeating the whole stuff with better code. As I told you, we are also developing the code, and it was not the methodology that improved, it was our code that improved.
I see.
Now the question that you are asking, we are asking ourselves, but this is now work in progress, and I agree, it's a very interesting one. We're gonna get back soon.
Yes, because I recall now with our colleagues from Canada, Roman Gordon, who has a interesting, I would say, a unique technique to probe the oscillations of single proteins trapped in a one optical device, and they made interesting molecular dynamics simulation paper where they showed when they have a nanoparticle in the water solution and they drive it just by embedding the forces but directly to the basically it was non-equilibrium MD and just driving basically the motion of the particle. They could show that at certain range of the amplitudes, the surrounding water molecules were basically not able to follow the oscillation of the particles, and they behaved as if they were closing kind of effective small vacuum gap. So it's a very fair description.
That's really interesting because here, when we have this buckyball, right here. When you want to, when you compute, I said that we determine the slip length and so on. So to that end, you need to know what your hydrodynamic radius is. And the hydrodynamic radius is quite tricky, actually. So what, here I remember it was like radius plus one layer of water around. And it's not clear what it is, actually. And basically something that you put into the continuum equation to act like radius, but this is not just radius of this buckyball, it's a little bit more. And it means actually what you say, but here we assume that there is a layer that sticks together to the buckyball, and it basically effectively enlarges the radius of the buckyball that the water is. Now what you are saying is this is really interesting because basically this would explain, because here we didn't explain this fact. Okay, here we just say, okay, our results and everything shows that we have one layer of water around the buckyball, and this together forms the hydrodynamic radius, but we didn't say what these molecules are really doing there. So if you say that this really, that there were people studying this kind of behavior in this first hydration or shell, I don't know where, that would be, that could actually determine many of these things here.
Okay, so I'll send you the paper.
Slip length definition and so on,
I'll tell you the paper because it's very interesting, because that would be, if this is the case that molecules don't follow, it's basically it's kind of corresponds to some kind of simplified slip condition or increased slip.
Because at that time, to me, what was interesting was basically, you know, when you are having a polymer, not a spherical object, a polymer, there are formulas how to calculate the radius of hydration. So there is an explicit formula, blah, and you have it. But here for the spherical, you don't have a formula. What is the hydrodynamics radius? It's not the radius of the molecule. Yeah. And nobody, so there were some discussion at the time. So you see that this is from 2012. They really didn't follow up to so much. But what these were questions that I was asking myself at that time. So basically here we just show we really did it in an iterative way. So we computed basically the velocity profiles, which you can always do. This is something of an observable. The velocity profiles of the water flow or water depending on the distance from the center of mass. Okay, so you had like a water profile, and then it was not zero and boundary and so on. So you could do that, and then you fitted this Stokes formula, okay, for the partial stick. But in this Stokes formula, as I told you, there you have this prefactor 4 pi, and then you have some fractional something that depends on slip length, which is the unknown, times RH, which is also the unknown. You have basically two unknowns.
I see.
Then it depends what you set for, you know, it depends the definition, what you will get out. Then you fit with this, basically the forces that you can measure. This you could also measure from the particle description, but also, so we use also the velocity profile in order to have like two different measurements for two different unknowns. Okay, so it was like, and then it's a little bit iterative, and then you end up with a good enough, you know, solution. But this does not explain, and out of that solution it comes that basically in our case this RH is radius of the buckyball plus one layer of water molecules. Okay, this is what we got, but this was a result of this fitting procedure, which is really engineering. So, but I would, if you want to really understand what's going on there, and I guess what you are just saying makes a lot of sense to me.
Okay, good. Thanks a lot. It was a very interesting discussion, and I think it would be very useful to keep in touch. I might send you some of these papers just to give
absolutely,
feedback on that.
I would be very grateful. Yeah, and yeah, absolutely, we should keep in touch. So it's good if you know, when you enter a new field, you don't know the people. So sometimes it's good to know new groups, and a lot of new ideas may come out. Yeah, I agree.
Great. Thanks a lot. I need to conclude to move to the also other meetings I have. But thanks a lot for very interesting discussion. And well, yeah, I will keep you in the mailing list, and we might come up. There will be some more, let's say, sub-Third spectroscopy talks coming in. So we'll keep in contact. I hopefully can leverage what we could exchange. Okay, Matej, so thanks a lot again for a great talk.
Yeah, thank you for the invitation, and I would be grateful as we keep in touch. Yeah, sorry.
We'll definitely do. So have a good day, and thanks a lot again.
You too. Bye bye.
Bye bye.